In this lecture, we'll be discussing amino acid metabolism. And on the title slide here, it's shown the nitrogen cycle in the environment. And it just helps a little bit explain, why it's important for, like in our cells, to metabolize and recycle the nitrogen. Because all the nitrogen on the planet that's available to us comes from the air, and we can't directly take the nitrogen from the air. We need these special bacteria that actually undergo nitrogen fixation to bring that from the air into more usable forms that then can be assimilated by plants and then indirectly by us through the plants. Okay. And the material in this lecture will be from chapters 18 and 22, but I'm going to go in a little bit of a different order. I'm going to start with chapter 22, section 1, then we'll cover chapter 18, and then finally chapter 22, section 2 at the end. And I think that just makes a little bit better story, more cohesive to do it in this order. Okay, so chapter 22, section 1 is an overview of nitrogen metabolism. Okay. So why is it important to do this, to undergo metabolism both catabolically and anabolically with amino acids? It's because nitrogen is scarce. Okay, so when we look at the actual breakdown and building up of some of these amino acids, just keep that in mind. The logic that these reactions are using is that nitrogen is sort of a scarce element biologically, so you need to conserve it as much as you can. And so the main this figure here, it's pretty good at explaining the different forms of nitrogen available biologically. You don't need to know this, all of this. What I wanted to point out with this is that the organic nitrogen, or N2 gas, right? That's very prevalent on the Earth in the atmosphere. It's about 80% of the atmosphere, but we can't use it. Okay, so we're really dependent on organisms that can take that nitrogen and fix it into either NH3 or other forms like ammonia. Okay. From there's other bacteria that undergo what's known as nitrification, and they can turn it into different nitrates or nitric oxides. Then those can be assimilated by plants, and then we can use those nitrogen groups by ingesting the plants. Right. And some of our activities as humans does challenge this nitrogen balance. One, we use a lot of fertilizers which add extra nitrogen into the biosphere, and then non-farming manufacturing actually does also release additional nitrogen into the atmosphere. So we do add more nitrogen into the biosphere ourselves as well, which then has to be it puts a stress on that whole cycle. Okay. Well, nitrogen, how it's fixed biochemically is by an enzyme, a complex enzyme known as nitrogenase. Okay. Only certain bacteria in archaea have this enzyme, right? And they're known as diazotrophs. Okay. And some of them are listed here. Not really going to talk about them individually, just sort of overall as a group. Okay? And kind of the scary thing is with more pollution and stresses on the environment, it can put these at risk, and there's a relatively small number of organisms that can do this. But life as we know it on the planet is really dependent on them, right? Because if we don't have nitrogen fixed, there's no way for us to make proteins, and if we can't make proteins, we can't live as we know it, right? So these are very important, and any pollution or something like environmental change that could harm these and maybe make them go extinct or just put them in danger really threatens the balance of life on the planet. The fixing of nitrogen, the reaction that occurs is this one down here. Okay, it's also industrially known as the Haber process. You're taking nitrogen gas and hydrogen gas and combining them into ammonia. It's a very exergonic reaction, so the delta G's negative, so this is a spontaneous process, but it doesn't occur very, very rapidly, so it needs to be catalyzed, right? And the reason it doesn't occur very rapidly here is because it has a very high activation energy. The overall reaction, if catalyzed by the nitrogenase enzyme complex, is shown here, and you see that for one nitrogen molecule to be converted into 2 ammonium molecules, it requires 16 ATP, right? So that's a very high energy cost to do this, and that just illustrates the high activation energy this reaction has. Remember, the delta G is quite negative, so it should be spontaneous, but yet this enzyme still has to use a boatload of ATP to do this, to overcome that activation energy. The enzyme looks something like this. Okay, you don't need to know this structure. What it does have here are some cofactors. Iron-sulfur center, which we've seen before. Electrons are then transferred to what's known as the P cluster, which is a little bit more complex. And then through the iron-molybdenum cofactor, FeMo. And then finally to nitrogen itself. Okay, so it uses sort of a pattern of electron transfer through these metal cofactors that we've seen before in respiration. They're just a little bit different, as you might expect. The redox, what am I trying to say? The reaction needs to flow downhill, this electron transfer, so they just have to be tuned to the correct reduction potentials. All right, this enzyme complex is actually unstable in the presence of O2. So what does that mean? That means that the bacteria that use this nitrogenase complex, they have to be in fairly anaerobic environments so that O2 is not present. So this reaction can occur. Right, and that's why most of these live in the soil, a little bit deeper in the soil where oxygen is scarce. And one particular type of bacteria that does this has a symbiotic relationship with the legumes. So like plants like beans. These bacteria live in the roots, nodules in the roots of these plants, and they form this symbiosis. Right. The bacteria benefit from having access to a large reservoir of energy in the form of carbohydrates and citric acid cycle intermediates from the plants. And they're also being bathed in a solution of oxygen-binding heme protein, this protein here that's abundant in these legumes. Right? So that removes oxygen pretty readily, so that it can be anaerobic, and then they also have a large store of energy from the plants. The plants benefit from this because these bacteria are supplying a huge amount of this fixed nitrogen to them. Okay, so there's this symbiosis with these plants. So once nitrogen is fixed in the form of ammonia, it can be incorporated through biomolecules, primarily through glutamate and glutamine. And so this is kind of a theme we're going to talk about in this lecture with alpha-ketoglutarate, glutamate, and glutamine. And I just wanted to point out here, they all have 5 carbons. They're all 5 carbon chains, right? There's a little bit of difference between them, and it's the number of nitrogen groups or amino groups each of them has. So alpha-ketoglutarate has no nitrogen groups. You add one of them, one nitrogen group to it, and it becomes glutamate. If you add a second nitrogen to it, it's glutamine. Okay, so alpha-ketoglutarate, 0 amino groups, glutamate, 1, and glutamine, 2. And that's kind of something we'll come back to. The enzyme that synthesizes the transformation between glutamate and glutamine, glutamine synthetase, is shown here, and this is a major regulatory point in the biological use of nitrogen. And you can see that things like glycine, alanine, histidine, tryptophan, those are amino acids. In we'll see near the end of lecture in the biosynthesis pathways, those are all members of other families besides glutamate. And if they're in abundance, they will inhibit this reaction. Okay. Other things that are sort of direct downstream products from that are available from glutamine, and they all work sort of in combination to provide this cumulative feedback inhibition. And we'll see this figure again at the end of the lecture. Okay, so moving to chapter 18 now, the metabolic fates of amino groups. Well, amino acids can undergo oxidative degradation when amino acids are released during protein turnover, so then they're not needed for new proteins, so they can be degraded and used for energy. Amino acids that are ingested that exceed the body's needs for protein synthesis, those excess amino acids can be broken down for energy. And then cellular proteins can be used as fuel if you don't have carbohydrates available. And let's give this figure, we'll just start here with the example of a protein that needs to be degraded, or a dietary protein coming in through ingestion is degraded into amino acids. We then have these free amino acids around, and what happens to them, they get deanimated, so the amino group gets removed. The carbon skeleton from those amino acids can then be fed through the citric acid cycle for energy. The amino groups can go into the biosynthesis of either other amino acids, nucleotides, or some biological amines. Okay, if those are not needed, amino groups in the form of ammonia or ammonium, they're actually toxic to be freely flowing around in the bloodstream. So they need to be excreted if they're in excess. You don't want them floating around, and so that's where this urea cycle comes in, and we'll be discussing that next section. This figure shows just a good overview of amino group, the catabolic process to remove amino groups. So we have, we'll be explaining a little bit more about each of these little steps, but I think I'd go into a little bit of detail here. In this sort of brown box here is representing a liver cell. Okay, so what you have is you don't want your ammonia groups or ammonium groups floating around in your bloodstream. So what happens is if you have from other tissues excess ammonia, how it gets transported is either by glutamine, so that comes from muscle or other tissue. Alanine is a big one to transport from muscle. And then you can have free amino acids flowing through the bloodstream from dietary intake of protein. Okay, so that's how it will get to the three main ways it gets to the liver. Okay, once inside, the fates, they all eventually get turned into ammonium. Okay, and then that can be excreted depending on what type of organism you are. You can excrete it differently. How it gets interconverted here is also shown in the figure. So when glutamine comes in, right, glutamine can just lose one of its amino groups, right, and that can be excreted. When it does that, it turns into glutamate. Okay, amino acids, dietary amino acids that come in, or alanine that's coming in from the muscle, they both can be converted. Their amino groups can both be removed using alpha-ketoglutarate. Okay, alpha-ketoglutarate can take the amino groups from either of those types, right, and become glutamate. Okay, so you're adding one amino group to alpha-ketoglutarate to become glutamate. What's left over from both of those, either it can be an alpha-keto acid going to the citric acid cycle, or if you're alanine, if you take that amino group off, you become pyruvate, right? And then that amino group that you've transferred to make glutamate can then be transferred back to ammonium to be excreted, right? And when you do that, glutamate becomes alpha-ketoglutarate again. So this glutamine, glutamate, and alpha-ketoglutarate are important for this process. And when we're talking about excreting nitrogen, you can be one of three. If we're talking about specifically animals, those that excrete their amino as ammonia, these tend to be mostly aquatic species, and that's because the ammonia gets diluted. It's toxic, remember, but if you're in water, you live in water, it gets diluted very rapidly, so you can excrete it as that. There's animals that excrete nitrogen as urea, so that's most terrestrial animals do this. And then those that excrete it as uric acid, and that's primarily birds and reptiles, right? Uric acid is not very soluble, so that makes it, I guess, harder to excrete as we would normally think of it, right? That's why birds don't really pee, but it's excreted at more of a solid form. Well, I'm going to include the next few slides. You're not going to be responsible for this information in this class. I think this is kind of more of a physiology description, but I think it helps to understand how we ingest foods, the proteins that we ingest with those foods, how they are broken down into amino acids, which then can be transferred through the bloodstream. So just going quickly through this, when you eat something, this hormone gastrin is secreted in the stomach, and it stimulates the secretion of hydrochloric acid and this pepsinogen. Okay, pepsinogen is what's known as a zymogen, which is a fancy word for an inactive form of a proteolytic enzyme. So an enzyme that breaks down proteins is called a proteolytic enzyme. And a zymogen is an inactive form of it. And so you when it's stored in those stomach cells in the stomach lining, you don't want that to be active because it'll just be breaking down the proteins inside those cells. Okay, so you want it to be in an inactive form, then it can be secreted and then turned to an active form. Okay, and that's just done because there's a very low pH when you have HCl in there in the stomach. The low pH will activate this pepsinogen to what's known as pepsin. That's the active form. Okay, and then HCl and pepsin can help break down proteins in the stomach. This process continues on into the small intestine. Okay, secretin is a hormone secreted into the blood in response to this low pH in the small intestine, and that stimulates the pancreas to secrete bicarbonate into the small intestine. Okay. And then some other proteolytic enzymes are secreted by the pancreas. Okay, that's trypsinogen, which is the zymogen of trypsin. Okay, chymotrypsinogen, which is the zymogen of chymotrypsin. And then those carboxypeptidases A and B as well. Right, so the accumulation of all of those enzymes will break down proteins into individual amino acids. Right, and when they are broken down into individual amino acids, they can be transferred into the epithelial cells that line the small intestine and then enter the bloodstream through the capillaries. Right, and thus travel to the liver. Okay, so that's how we're getting some of these free amino acids into the liver for further metabolic processes. Okay. The ones that are really involved, as we've seen, glutamate and glutamine, those are involved in this process of moving nitrogen around through alpha-ketoglutarate. Okay, some other important ones, alanine to pyruvate, which we've sort of also seen that one on that overview slide. What we haven't yet seen is aspartate, and aspartate works through oxaloacetate. Okay, and remember, oxaloacetate is something we've seen before in the citric acid cycle. So the way that the nitrogen is transferred biologically, it involves a cofactor called PLP, or abbreviated as PLP. That's the abbreviation for pyridoxal phosphate. Okay, and it's a cofactor, and you might remember it from some enzymes we've talked about before, but we'll go through it a little bit more here in a second. These enzymes are sometimes referred to as aminotransferases, which is a bad name. It's a bad name because it sort of implies that it's just taking an amino group and sticking it on something, which is not really the case. The older name, transaminases, is better because that's sort of telling you it's taking an amino group from some other molecule and moving it to another molecule. Okay, so and these reactions all require a donor for your amino group. Okay, they're not using free amino groups, right? Because remember, free ammonia or ammonium would be toxic. So these amino groups are on some other molecule. And so the classic one that we've been using here is this ability to take an amino acid and alpha-ketoglutarate, right? You move that amino group from an amino acid group and put it on alpha ketoglutarate. What you get is glutamate, and then you get that alpha keto acid of whatever amino acid the amino group was starting with. So you're just transferring that amino group to alpha ketoglutarate to make glutamate, right? And so alpha ketoglutarate, glutamate, and glutamine are very important in this process. Right, so these transamination reactions all require, if you have an alpha amino group, you're transferring them to the alpha carbon of alpha ketoglutarate to yield your alpha keto acid analog of the amino acid. All right, and the delta G of these are all pretty close to 0, which means they can be reversible. So you can use them to break down amino acids as well as to synthesize them. Okay. And just another way of looking at it is shown here at the bottom. Right, you have various enzymes that catalyze these depending on which amino acids you're starting with. Okay. You can use, they all tend to use alpha ketoglutarate and glutamate for their acceptors, right? And then glutamate can then be broken down back to alpha ketoglutarate using glutamate dehydrogenase, which actually reduces NAD to NADH. So that cofactor we were talking about that's important in these reactions is pyridoxal phosphate, or PLP. It is a form of vitamin B6, so an active form of vitamin B6. So you take in B6, this is ultimately what it's going to be used for. Its forms, the PLP form, the pyridoxal phosphate form, is the form that is going to be accepting the amino group. So that's the aldehyde form. Right, so what it is a 6-member ring. It does have a positively charged nitrogen in that ring and a phosphate group attached to it. But this is the point of attachment that this cofactor uses. So the aldehyde form is what's going to accept that amino group. Once it's accepted the amino group here, we get what's known as pyridoxamine phosphate. Okay, so pyridoxal phosphate is the form that doesn't have the amine group. When it attaches an amine group, it's pyridoxamine. Right. Here's some other reactions that are facilitated by pyridoxal phosphate. One is transamination, which we just talked about. You can get racemization, which is turning let's say an L form amino acid into a D form. Okay, and then the other is decarboxylation. And we've talked about some of these decarboxylations, specifically pyruvate decarboxylase, which we've talked about before in this class. But there are other decarboxylations that can occur. One that's shown here, what are we using? I guess it's just a generic amino acid it can use to decarboxylate that. The glycine cleavage enzyme that removes CO2 from glycine and breaks glycine down uses pyridoxal phosphate. Okay. So glutamate ultimately releases its amino group as ammonia in the liver. Okay, so all things sort of funnel through glutamate, and glutamate can then release that ammonia group in liver. And how that works is this enzyme glutamate dehydrogenase. So it's catalyzing that oxidative deamination of glutamate to produce ammonium and alpha ketoglutarate. Right. So the interesting thing here is it can use either NAD or NADP, right? So it can use either. It's oxidative because you're oxidizing your glutamate to alpha ketoglutarate, and by oxidizing that, you're reducing that cofactor, right? And then those can be, fed into things like the main respiratory chain. Alpha ketoglutarate can, either be used for energy in the citric acid cycle, or it can be used for glucose synthesis ultimately, right? So this is an important intersection in carbon and nitrogen metabolism, right? You're generating alpha ketoglutarate in nitrogen metabolism, and that then can go feed into carbon metabolism, either in the form of gluconeogenesis or citric acid cycle. Right. Some things that glutamate dehydrogenase, it's positively modulated by ADP. That tells you that there's need for energy, right? If you have a high ADP compared to ATP, that would signal low glucose levels. It's negatively modulated by GTP, and that just signals high levels of alpha ketoglutarate, and that you don't need to be breaking down your glutamate. Right. And as we've mentioned, ammonia is toxic to animals, and especially in the brain. And a couple reasons why that is shown here. These transporters are all affected by ammonium ions, so the ammonium is sort of competing for these transporters, and you can get, kind of messed up concentrations of ions, extracellularly or intercellularly, and affect the transport, and then that then affects the things like the response to neurotransmitters. So that's why it's important to remove your free ammonium. Right? And glutamine is one way that you can transport this ammonia in the bloodstream. And how we make glutamine, we can start with glutamate, okay, and we add a free ammonium group to that. Okay, and that's done in a two-step process by glutamine synthetase. Okay. First step, we're activating our glutamate by sticking a phosphate on it. So we're using ATP to do this. Okay, and so ATP is pretty precious, right? But we need to use some of it to get rid of this toxic component of metabolism, this ammonium group, out of the cell. Right. So first we activate glutamate, then we can attach the ammonium to it to make glutamine. Okay. The opposite reaction, once glutamine reaches the liver, is by this glutaminase. Okay. Glutaminase catalyzes the reverse conversion to take that ammonium group off, and we get our glutamate back. Okay, glutamine, 2 ammonium groups. Glutamate only has 1. That extra 1 is now ammonium, and it can move through to the urea cycle. All right. So that's glutamine is used to transport from most tissues. Muscle, how it transports, or a major way in which muscle transports ammonia groups is through alanine, the amino acid alanine. Okay. Right, and that's because skeletal muscles are producing pyruvate, lactate, and ammonium. And they use this alanine aminotransferase to interconvert pyruvate and alanine via this transamination reaction with glutamate. Right. So if we're looking here in muscle, right, muscle is taking glucose and we're going through glycolysis to make pyruvate for energy. Okay. Muscle protein can be when this is happening, rigorous exercise, you can be breaking that down, your muscle protein, and producing, free amino acids. Okay, when you do that, ultimately you're going to be producing ammonia groups. If you combine, a glutamate, one of those amino groups can be transferred to pyruvate that you're producing through glycolysis, right? And then you go this transaminase reaction here through alanine aminotransferase. And what happens is pyruvate will accept the amino group and become alanine. Okay, glutamate gives it up and becomes alpha-ketoglutarate, right? So we have this cycle here with glutamate and alpha-ketoglutarate again, right? So pyruvate becomes alanine. We can now transport alanine through the bloodstream because it's non-toxic. It goes to the liver. And in the liver, the reverse reaction happens with alanine aminotransferase, where we use alpha-ketoglutarate to accept the amino group from alanine. So it becomes glutamate. Alanine goes back to pyruvate. Pyruvate then can go through gluconeogenesis to become glucose again. Glucose then can flow through the blood back to the muscle where it's needed. Okay, so this is the glucose-alanine cycle between muscle and liver. Okay, so how do we get rid of the nitrogen, the excess nitrogen that's available? And that's through urea cycle in human and most terrestrial animals, right? So this is a very small figure of the urea cycle here. Probably hard to read on this slide. It's in your book. One thing that this figure does show very well is that one of these steps, the first step into this pathway, is in the mitochondria. The other steps in it are in the cytosol. So I just thought I'd point that out. Okay. Right, so this is the pathway which the ammonia deposited in the mitochondria of liver cells is converted to urea. Okay, and urea enters the bloodstream and it's excreted into urine. Okay, and that's how we get rid of it. So we turn our ammonia groups into urea because that is a less toxic and more easily transferred molecule to get rid of the nitrogen. All right, so let's look at these steps individually. Okay, the first step, we're taking ammonium and converting that into a compound carbamyl phosphate. Okay. And what you'll notice here is there's one carbon in it, a phosphate group, and your amino group. Okay, so what this enzyme needs is bicarbonate, 2 ATPs to do this reaction. Okay. And this is the one reaction, the first step, and it's recurring in the mitochondrial matrix. Right. So that's we can really think of that as the entry point into this cycle, because we just describe this as a cycle, which means there's technically no beginning or no end. But how the amino groups go into this, they get turned into carbamyl phosphate. Okay? Then they can start the first step in the cycle, right? And that's catalyzed by ornithine transcarbamylase. Okay, so we're taking ornithine, which is an amino acid, by the way. It's just not one that's used in protein synthesis. And it's turned into citrulline, which is also an amino acid. Okay, what you're doing here is you're taking this group that's shaded yellow and blue, and you're just sticking it on the end of an ornithine. Okay. All the other steps now are going to be occurring in the cytosol. Right, so the next step, citrulline goes into the cytosol, transferred from the mitochondria to the cytosol, and then we have this argininosuccinate synthetase enzyme that does this two-step process to make argininosuccinate. And what we're doing, it's condensating the amino group of an aspartic acid or aspartate and that ureido group of citrulline to form this argininosuccinate. And it's shown here, right? Aspartate is colored sort of this orangey and green to show the amino group of it. And it's just stuck onto our citrulline. And it goes through this intermediate. Okay, not important that you know the intermediate it's going through. We're using ATP again here. Okay, so that's it's sort of another sign that shows you how important this is, that we're using energy to get rid of excess nitrogen. Okay. Well, we can use our argininosuccinate now to form arginine, and doing this, the enzyme is called argininosuccinase. Okay, we're doing that reaction. What we're forming out of it is also a fumarate molecule. All right, and then finally, final step of this cycle, we have arginine and an enzyme called arginase that cleaves this piece off, the colored piece off here, of arginine to form ornithine. Okay, and that little colored piece that it cleaves off is urea. And you'll notice that urea has two nitrogen groups and a carbonyl, a ketone, in the middle of them. One of those nitrogen groups is the one that came in from the start in the mitochondria. And then that can be excreted. So that's a way that we're excreting nitrogen groups ultimately. All right, this urea cycle is actually linked. It's linked through to the citric acid cycle, not directly, but through that argininosuccinate. If you remember, that compound was turned into arginine, and a molecule of fumarate was also created when we did that, right? Well, fumarate can be turned into malate. Malate can be shuttled back into the mitochondria, and that's the entry point into the citric acid cycle. So these are linked, okay? Every turn you do of the urea cycle, we're creating a fumarate that goes into ultimately back into the citric acid cycle. All right, and that's what's known as the aspartate argininosuccinate shunt. And that's the pathway that links both the citric acid cycle and urea cycle. Right, these, if you think about aspartate now as a nitrogen donor, the pathway for nitrogen incorporation is one of the two common ways to introduce amino groups into biomolecules. The urea cycle and citric acid cycle, they're closely tied together, and that brings NADH in the form of reducing equivalents into the mitochondria, right? And why that's important, that can be used to generate ATP. Right, so this pathway is pretty ATP intensive, right? We've seen in the urea cycle, we're using ATP in a couple of those steps, right? So to offset the cost of that, being tied to the citric acid cycle actually helps, right? That creating of the fumarate that then goes in the citric acid cycle will help produce ATP, so it helps offset this cost. Okay. Right, so the next couple sections, we're going to talk first about how amino acids are broken down, the catabolic processes that break down amino acids, and then in section 22-2, we're going to talk about how they are synthesized, so how they're built up, or the anabolic pathways. Okay. I will mention that we're just going to be taking a gross overview of this. You're not going to be responsible for the very detailed, chemical steps that each of these have. They are very complex. I work in this field, and for me, I still don't know most of these pathways. Right? Because I'm just kind of focused on one sort of small pathway or a couple of them. So it's I can't expect you to memorize these. They can be very complex. In reality, they're all sort of interconnected with other things, and it gets very complicated. Let's just leave it at that. So amino acid catabolism, it's not a very large for the production of energy, right? It's much more efficient to get energy from sugars or fats, right? Carbohydrates or fats for the cell, but it still does provide 10 to 15% of human energy production. And you can imagine, right, just the extra cost of dealing with those free amino groups is challenging, and the fact that those amino groups and biologically are sort of scarce, so you really want to recycle it as much as you can. That's probably one of the reasons why it is not such a big energy producer. Okay, but it still does account for some energy production. So there are listed here 20 catabolic pathways that converge to form 6 major products, right? And those major products are the ones that you should focus on, not necessarily the individual amino acids, but just what they tend to turn into when they're being broken down. Okay, those are pyruvate, acetyl-CoA, alpha-ketoglutarate, succinyl-CoA, fumarate, and oxaloacetate. And all of those we've seen before in glycolysis and the citric acid cycle. Right. So the summary here, and this is kind of like probably the most important slide of this section. So this is the one that you should really focus on, that you'd be kind of responsible for, is just knowing which amino acids turn into which of these 6 major molecules. Okay. And we'll go through all of these sort of kind of individually, each of these points. The one thing that you'll also notice on this slide is the different color codes. So that kind of orangey color here, these are considered glucogenic amino acids, and the blue ones are ketogenic, right? And they're ketogenic because they can be turned into, through acetyl-CoA or acetyl-CoA, they can be turned into ketone bodies, right? All the other ones are glucogenic because they can really be turned back into glucose, okay, through oxaloacetate. Right, and that just kind of explains what I just mentioned there. The ones that are ketogenic, right, those are things like phenylalanine, tyrosine, isoleucine, leucine, tryptophan, threonine, and lysine. Right, so if you're on a ketogenic diet for some metabolic problem, those are the amino acids that your diet would be high in. But several cofactors play important roles in the catabolism or breaking down of amino acids, okay, and those are sort of listed here, right? what they're doing is they're transferring. These are all involved in transferring 1 carbon units. So biotin, which is shown here, tetrahydrofolate, which is this big molecule here, or S-adenomethionine, shown here. Okay, they're all used for 1 carbon transfers. Tetrahydrofolate, we'll go just show a little bit more detail of. It's a larger molecule, and I guess the more important part is shown here. What it's actually synthesized in bacteria, and it's referred to as folate, and there are different forms of it. Okay, those forms are shown here, and you can have methyl folate, methylene folate, where are we? Formal folate. And then just the formate, which would be the absence of the carbon group, right? So all of these forms are either more reduced or more oxidized. Okay, so the most reduced is the methyl, the most oxidized is the formal. Okay, and just coincidentally, I am currently trying to create a LC-MS method that can quantitate all forms of these. It's very challenging though, because they are all, most of them are sort of unstable and they can be interconverted between each other. So it's a pretty challenging process. Right, so those are the cofactors that are typically used by these enzymes. We're not going to talk about any of the enzymes specifically though. Okay. What we will talk about is what amino acids are degraded to which entry point products. Okay, so pyruvate, there are 6 amino acids that are degraded ultimately into pyruvate. Those are alanine, tryptophan, cysteine, serine, glycine, and threonine. Right, so these are converted, we can think of them converted into pyruvate. Okay, pyruvate then can either be converted through pyruvate, the PDH enzyme system, into acetyl-CoA and go into the citric acid cycle, or oxaloacetate can be used to go to gluconeogenesis. You're not responsible for any of these specific reactions or enzymes. I'm just including them in the slides in case you're curious. I don't know if these would show up on the MCAT. I sort of doubt it, but it might help some of you remember how these are related, but you're not responsible for any of these specific reactions. Okay. Asparagine and aspartate are 2 amino acids that are degraded to oxaloacetate. And I suppose those are, asparagine and aspartate are sort of related, if you will. So keeping track of those might be easy. All right, 7 amino acids are degraded to acetyl-CoA. Leucine, lysine, phenylalanine, tyrosine, and tryptophan are first converted to acetyl-CoA, which then can be turned into acetyl-CoA. Isoleucine, leucine, threonine, and tryptophan can be converted directly to acetyl-CoA. And you'll notice that there are some overlap here. Like tryptophan, for instance, is listed in both of these. So it can, its degradation pathway could go either way. Right, so there's some overlap. Hey, those specific, each step specifically shown here, again, not required to know that. Tryptophan, it's very complex. maybe not surprisingly, it is a very complex side chain there, but it can be broken down into a number of different intermediates that can be precursors for other biomolecules, and some of those are shown here. Things like serotonin or the piece of the NAD or NADP cofactors. 5 amino acids are converted to alpha-ketoglutarate, and we've already talked about a couple of these, right? Glutamate and glutamine. Okay, the others are arginine, histidine, and proline, right? They can be, these 4 can be converted into glutamate. Glutamate can then be converted into alpha-ketoglutarate. Right, and specifically those, the pathways are shown here. Right, 4 amino acids converted to succinyl-CoA. They're isoleucine, methionine, threonine, and valine. And some of these conversions of branch chain amino acids, so specifically when we say branch chain amino acids, we're talking about valine, isoleucine, and leucine. Those are actually not degraded, not carried out in the liver. There are other tissues such as muscle, fat tissues, kidney, and brain, or where these branch chain amino acids are converted, which I thought was just kind of interesting. That branch chain alpha keto acid dehydrogenase complex is what does this breakdown of these branch chain amino acids, and it does an oxidative decarboxylation of all 3 of these, releasing CO2, producing the acyl-CoA derivatives. All right, and so that should look very familiar to the alpha-ketoglutarate dehydrogenase complex in the citric acid cycle, which we've discussed before. Okay, it's virtually identical except for the active site, binding site for the molecule. It just fits these branch chain amino acids instead of alpha ketoglutarate, right? And goes through the same mechanism, and very closely related to pyruvate dehydrogenase as well. If you have a problem in this complex, you get what's known as maple syrup urine disease, and I thought I'd bring that one up because in our clinical laboratory at Anschutz, we do this is one of the diseases that we do see, and it has a very unique name because of the specific smell that the urine gives off when you have this disorder. Right, some other genetic defects in amino acid metabolism are shown here with their approximate incidence per 100,000 births, and you can see that they're all, fairly rare. The phenylketonuria shown at the bottom is probably the most common, I would say, and that's the inability to convert phenylalanine to tyrosine. Right, if you have that defect and you ingest a lot of phenylalanine, like in the form of like a diet soda, something with NutraSweet in it, has a lot of that phenylalanine in there, you can stress your system and bad things can happen. Okay. So if you're interested, these are just some of the amino acid disorders that you can if you go into this field that you can run into. Right. So that was the breakdown of these amino acids. The last thing we'll talk about is the biosynthesis of amino acids. And what you'll notice is it's a little bit different. So how they're broken down and how they're built back up is not always the same. So it's slightly different pathways. And again, just as in the case of the catabolism, for these anabolic pathways. I'm not going to expect you to know any of the real details. I just want you to know what we'll call the families. There are 5 families of related amino acids for biosynthesis. Okay. So the overview is shown in this figure, and what you'll notice is they're all stemming from either glycolysis or the citric acid cycle, right? There are exit points on both of those main central metabolic pathways, exit points from those that can be used to synthesize amino acids. Right, so you'll see things like 3-phosphoglycerate can be turned into serine, so that will be one family. Glucose 6-phosphate can be turned into ribose 5-phosphate, which then can be turned into histidine, so that's another. 3-phosphoglycerate can be turned into erythrose 4-phosphate. That and PEP, phosphoenol pyruvate, can be used to make the aromatic amino acids, so that's another family. Pyruvate can be used to make these amino acids. Alpha-ketoglutarate, as we've seen, can be used to make glutamate, glutamine, proline, and arginine. That'll be a family. And then oxaloacetate through aspartate into these amino acids will be yet another family. Okay. Well, organisms actually vary in their ability to synthesize amino acids. And there are 20 that we consider the common amino acids, the ones that are involved in protein synthesis. If you're a plant or a bacteria cell, you're able to synthesize all 20 of these common amino acids. Mammals can synthesize only about half. Okay? The ones that we can synthesize, we consider non-essential amino acids because we don't need to take them in from our diet. The others that we can't synthesize, we call essential amino acids. And so it's hard to really give a number here, which ones are really essential, because that number can vary depending on the source you look at. It also varies on whether you're a child or a grown adult. Some of them can change. Probably sort of varies a little bit on what tissue you're looking at. So we're not really going to try to classify them exactly, but just know that about half of them are considered essential. Okay, what we will focus more on in the class is the biosynthetic families. And they're all summarized here in this table. Okay. There are 6, I think I said 5 earlier. There are actually 6 of them. The histidine one seems kind of funny to call a family because it's just a single amino acid, but here they're listed. Okay, and just going through them sort of quickly, but the alpha-ketoglutarate, or this is sometimes called the glutamate family, since alpha-ketoglutarate isn't technically amino acid. In the glutamate family, they all stem from alpha-ketoglutarate, right? And we've seen this conversion before, this relationship between alpha-ketoglutarate, glutamate, and glutamine, right? The other 2 that can be synthesized from glutamate are proline and arginine. Right, and you can kind of notice how different this is than the catabolic processes that we talked about. The families between biosynthesis and breaking down, the oxidative degradation, they're going to be slightly different, which makes it a little bit more challenging. Okay. The serine family all stems from 3-phosphoglycerate. Right, so you might want to call it the 3-phosphoglycerate family, right? That can be converted into serine. Serine then can be converted into glycine or cysteine. Okay, so those three amino acids are all related to each other. Right. This slide shows two families. One is the aspartate family through oxaloacetate. The other is the what we would tend to refer to as the alanine family through pyruvate. Okay. Aspartate can be turned into asparagine, methionine, threonine, and lysine. Pyruvate can be turned into alanine, valine, leucine, and isoleucine if pyruvate and threonine combine. So these two families here are sort of related because of isoleucine. The aromatic amino acids can be synthesized from PEP, phosphoenolpyruvate, and urethrose 4-phosphate. Okay, those coming together can make phenylalanine, tyrosine, and tryptophan. Phenylalanine can be converted into tyrosine as well, right? And that's that break point there is one of those disorders, the amino acid disorders, right? So, right, even though you can, if you can't synthesize tyrosine from phenylalanine, that can be an issue because you get a buildup of phenylalanine, right? And then finally, that lone histidine, it's in its own family. It's synthesized from ribose 5-phosphate, which is a intermediate in the pentose phosphate pathway. Right, and so all of these things sort of feed back into that glutamine synthetase enzyme, the one that takes glutamate and makes glutamine. Right, so that conversion of glutamate to glutamine, glutamine then can be used for a number of different things ultimately in the cell. But these amino acids here, when they're in high concentration, things like glycine, alanine, histidine, or tryptophan, and you'll notice that all of those are in different biosynthetic families, right? They can act to feed back in downgrade our glutamine synthetase, right? So this is sort of a net feedback, okay? And it's what it's called is concerted inhibition. Okay, that's regulation where products derived from biomolecule serve as a negative feedback modulators of the enzyme producing the biomolecule. And so this overall effect are more than additive. Okay, so it's not really like a simple process of just one pathway, but multiple pathways on this enzyme. Okay, so that concludes this lecture. Again, amino acid metabolism is very complex, and you could probably spend a whole semester talking about just this topic and going through all of these different pathways. But I wanted to provide this as more of an overview, just so you get the basics and get sort of comfortable with it. So really in this section, we'll try to focus more on the overview of it as opposed to the nitty-gritty details.